Hello, welcome to the continuation of our Chapter 13 lectures on viruses, viroids, and prions. This video will talk about viral replication in bacteria. So I've been showing this cartoon for viruses in all the video lectures. Here's the phospholipid bilayer of the host cell. That would be our cell. Oh, this is in German too. The Wirtszell is the host cell in German. And then here's receptors, ACE2 and TMPRSS2 sticking out of our host cells, specifically our respiratory epithelial cells. And then here's the virus. So this is coronavirus 2. Here's the RNA inside the coronavirus. These blue tulips are the spikes, and when the spikes, blue tulip spikes on the coronavirus fit receptors, specifically this ACE2 receptor on our respiratory epithelial cells, then the virus attaches to our host cells. And then also TMPRSS2 will also bind to these spikes and cause activation of the virus. So then the virus can get its RNA inside our host cells. Okay, so this is viral replication in bacteria. Excuse me, and then the next video will be viral replication in animal cells. So that would include our cells. So let's check it out. Here's a bacillus. That's the shape of this bacteria. It's a rod shape. And then remember, bacteria have this double-stranded circular chromosome. Unlike us, we have linear chromosomes. Bacteria have circular chromosomes, but that's where the DNA is. And here comes the lunar landing, bacteriophage. So bacteriophage is another name for a virus that infects a bacteria. So the first stage in this scenario is the bacteriophage will attach to the bacteria. So that's attachment phase. And then you can see here this bacteriophage is actually the nucleic acid from the bacteriophage is entering the bacteria, and the lunar landing module stays outside of the bacteria. And then in the third step, there are enzymes that come in with the virus that will degrade the bacterial chromosome. So remember, the bacterial chromosome was this purple circular chromosome. It got degraded, and now the phage DNA is the only DNA left in the bacteria. So its own DNA has been destroyed. Then the virus goes on to use the protein synthesis machinery in the bacteria to make its own viral proteins or phage proteins. So these are all the pieces of the phage, the virus that will be made inside the bacteria. And then there's the process of assembly. So all these pieces from the phage proteins will be assembled together, and then the bacteria will lyse or open up, and these new bacteria or bacteriophages are released from the bacterial cell. So one virus got in, but look, it used the cell machinery of the bacteria to make all these new infectious phages, aka bacteriophages. And here's the 5 stages: attachment, entry, synthesis, assembly, and release. So replication, viral replication, this whole process is viral replication, usually results in host cell lysis. That's this part of the cartoon here. The cell bursts open and then it dies. And this process of the bacteriophage getting its nucleic acid inside the bacteria cell, this is called lytic replication. Lytic replication because when the virus is done using bacteria, the bacteria lyses. So it's lytic replication of the virus. We've been saying this all along. Viruses cannot replicate on their own. They don't have ribosomes or any of the protein synthesis machinery, so they need to use the host cell. And in this cartoon, the host cell was bacterial cell. So once a virus has attacked, the host can't make its own proteins because, look here, the chromosome got degraded. So you can't take bacterial DNA and make RNA and then make bacterial proteins. Okay, so let's look at stage 1, attachment, in a little more detail. So here's a cartoon of this lower part is the bacteria. So it's got this outer membrane, and then here's its peptidoglycan, part of the cell wall, and then here's the bacterial cytoplasmic membrane. So do you know what kind of bacterial cell this is then? If it has an outer membrane, is it gram-positive or gram-negative? If you said gram-negative, you're right. Good job. And then here's a cartoon of the virus, or the bacterial virus called a bacteriophage, landing on the bacteria. And it just shows the bacteriophage, or the viral genome, is inside the capsid here. And then you have this piece called a tail sheath, and then here's the tail fibers. And really it doesn't show it here, but these tail fibers of the bacteriophage are recognizing some proteins on the bacterial outer membrane, so that allows the bacteriophage to attack and attach. So that there are attachment proteins on the tail of the virus, and there's receptors on the host cell outer membrane. Okay, stage 2, the entry of the virus into the host. So you can see here's the capsid of the bacteriophage. Here's the nucleic acid entering. So this would be the cytoplasm, or the intracellular fluid of the bacteria. So this is a specific phage called T4 phage. It releases lysozyme. So through here it releases lysozyme, and that degrades the peptidoglycan here in this layer of the cell wall. So there's an enzyme that will degrade that part of the cell wall. And then the phage's tail sheath, that's this part right here, it contracts, and that forces an internal hollow tube, this piece right here, into the bacterial intracellular fluid, aka cytoplasm, and then the DNA can gain entry to the cytoplasm of the bacterial cell. And then this empty capsid out here, that's left on the outside of the bacterial cell. Stage 3, the chromosomes are degraded. So bacterial chromosomes are degraded here. So the viral enzymes that came in with the viral nucleic acid degrades the bacterial DNA, aka chromosomes. Stage 4, so here we are with the synthesis. So protein synthesis is like regular cellular transcription and translation. So the DNA is transcribed into RNA, the RNA is translated into proteins. And so now the virus has used the bacterial host cell to just make a whole bunch of viral proteins, aka phage proteins. So you want to make, so these viruses or bacteriophages are making the head capsomeres. They're making pieces of the tail. They make viral DNA polymerase, and they make lysozyme. They'll be ready to go out into the world as virions. And then assembly of the viral particles. So they call it self-assembly because I think they don't really understand all the mechanisms. So they just say all these pieces of the virus. So the base, the tail, the sheath, the DNA, the capsid, the head, the tail fibers, all are assembled into this mature virion. And then the virion will be released in the next step. So we're right here. So once the virus has assembled all its parts to make mature virions, well, actually they'd be viruses at this point because they're not outside the host cell. Then the lysozyme that came in with the virus will degrade the bacterial cell wall here. The bacteria disintegrate, and the viruses are released. With this process, this lytic replication of bacteriophages or bacterial viruses, you can get about 100 to 200 virions per bacterium in about 25 minutes. You can make a lot. So that was lytic replication of bacteriophages. This is another way that bacteriophages can replicate, but this is lysogenic. Okay, so here's the lytic cycle. We just talked about this. This is lysogeny, aka lytic replication of bacteriophages. So it's got some of the same parts of lytic cycle, the lysogenic replication cycle does. So you've got step one, you have attachment. So the virus, in this case, they're using phage lambda as an example. It attaches to the bacteria, and then here the nucleic acid gets in. But check it out. Once the nucleic acid from the virus, this piece right here, is inside the bacteria, the host chromosome is not degraded. It's still intact. And so instead of destroying the bacterial chromosomes, the viral DNA inserts into the bacterial chromosome. So they call that now, the viral DNA is now called a prophage in the bacterial chromosome. And then check it out, the bacteria replicates. So every time the bacteria replicates, it's making more of this green piece here, which is viral DNA or a prophage. So this way, this is one tactic that viruses, bacterial viruses, can use to make more of themselves. Each time these bacteria replicate, you're just making more viral DNA. So you started out with this one piece here of viral DNA, and then through this process of lysogeny or lysogenic replication, now you've got, all these green pieces here. So you started with one, and now you have all these replicates of the viral DNA. And then at some point in this process, the bacteria go through this phase, the bacteria and the virus go through this phase called induction. And then at that point, the bacterial chromosome is destroyed, and now the viral or the phage DNA is left to just go ahead and synthesize its own parts. So then it goes here to synthesis, and then the parts assemble, and then you get the lytic release of the phages in the end. So some phages, or bacterial viruses, initially have or do lysogenic replication. And there are 5 stages of lytic replication that we talked about before. There's attachment, entry, synthesis, assembly, and release. That's the lytic replication. In lysogenic replication, they have these other steps. So, you have attachment, entry, then you have the prophage, then you replicate the chromosome that has the phage in it, and then you undergo induction, and then you go back into the regular cycle where you synthesize the viral proteins, you assemble the viral proteins, and then you release the whole virus. So lysogenic replication, obviously you have to have attachment. So you have to get the virus to attach to the bacterial cell. And then the nucleic acid, in this case DNA, enters the bacterial cell, but the host DNA is not destroyed. So remember, we said the host DNA here, that would be the bacterial chromosomes, not destroyed. So the phage or the virus doesn't immediately assume control, and it's called a prophage because it's inserted into the chromosome. It just lets the bacteria do a bunch of work by dividing and making more phage DNA. And then that's what I just said, the prophage inserts into the bacterial DNA. And this is interesting. at this point, when you insert that prophage into the chromosome of the bacteria, this could cause this process called lysogenic conversion of a bacteria. So the phage genes are here because it's the nucleic acid of the virus. They may code for a toxin. The bacteria may secrete that toxin, and that can cause diseases like diphtheria, cholera, rheumatic fever, severe cases of diarrhea. So that's when you convert to a pathogenic toxic bacteria. So really, bacteria was just going along minding its own business. It got converted to a pathogenic bacteria that releases toxins because of the insertion of the viral DNA in the form of prophage. And then this piece right here, induction, when the bacteria have divided many times, you can excise or take out that prophage, that viral DNA, from the bacterial chromosome, and then that bacterial chromosome disintegrates. Induction can happen because of physical and chemical agents that damage DNA. So we put a bacteria under UV light, X-rays, carcinogenic chemicals.